All right, so we're diving into some really fascinating stuff today.
Yeah,
Self-eliminating gene drives.
absolutely.
Um, you've given us a ton of great material to work with here.
Yeah.
Scientific papers, patent applications, research notes.
hmm
You've obviously been doing some digging into this cutting-edge technology,
For sure.
and that's what we're here for, to like really get into the nitty-gritty of it all.
But
So our mission today is to like explore this whole concept,
yeah.
delve into the science behind it,
Uh-huh.
the different organisms and mathematical models they're using,
Right.
and the implications for things like disease control.
Mm.
But here's where it gets really interesting.
Okay.
Self-eliminating gene drives are designed to disappear from a population over time.
Yeah,
It's like a self-destruct button for genetic modifications.
exactly.
So before we get too deep into the specifics, maybe we could just like level set for a second.
Sure.
For those who might not be as familiar with gene drives in general,
All
can you
right.
give us like a quick Gene Drives 101?
Yeah. Yeah. So, in the simplest terms, a gene drive is a way to make a genetic modification spread rapidly through a population.
Okay.
So, traditionally, if an organism inherits a particular gene, there's a 50-50 chance it'll pass it on to its offspring.
Yeah, classic Mendelian genetics.
Exactly. But with a gene drive, that probability is significantly increased.
Like turning the odds in your favor.
Precisely. And that's why they have such huge potential for things like disease control.
Right. You could theoretically engineer a gene drive that makes mosquitoes resistant to malaria
Yeah.
and then release those mosquitoes into the wild and watch that trait spread,
Mm-hmm.
potentially eradicating the disease.
That's the idea, but there have been some challenges.
Like,
Well, for one thing, there's the issue of control.
oh yeah, like once you release a gene drive into the wild, how do you stop it?
Exactly. Earlier studies using CRISPR-Cas9 have showed just how quickly these modifications can spread.
It's like hitting the gas pedal with no brakes,
Yeah, and that's led to some legitimate concerns about unintended consequences.
right? Like what if the modification spreads to non-target species or has some unforeseen ecological impact?
That's where self-eliminating gene drives or SEMs come in,
So SEMs are like gene drives with brakes.
basically.
Okay, I'm intrigued. How do they actually work?
Well, the PLOS genetics paper we have dives into the science behind it,
Uh-huh.
specifically something called the single-strand annealing pathway, or SSA.
SSA.
Yeah, it's a natural DNA repair mechanism that cells use to fix double-strand breaks.
Double-strand breaks, like when the DNA molecule literally gets cut in two.
Yeah, imagine you have like a blueprint with a tear.
Okay.
The cell uses a similar section of the blueprint as a guide to repair the tear. That's essentially what SSA does.
So researchers have figured out how to hack this system.
Yeah, they've learned to exploit this pathway to program the removal of transgenes.
Transgenes, those are like the artificially introduced genes, right?
Okay.
So they're basically engineering the gene drive to self-destruct.
You got it. They flank the gene drive with these specific DNA sequences called direct repeats.
Direct repeat.
And when a special enzyme called a nuclease cuts the DNA within the gene drive, the SSA pathway kicks in.
Okay.
The cell uses those direct repeats as guides, and in the process of repairing the break, it removes the gene drive.
It's like self-destruct instructions hidden within the gene drive itself.
Exactly.
That's pretty wild. And what's even cooler is that the patent application we have actually shows visuals of how they can manipulate the speed of this self-decay. It's amazing how much control they're gaining over this process.
Yeah, it highlights the level of precision they're aiming for.
So they can basically adjust the system to make the gene drive disappear faster or slower.
Yeah, depending on the application.
That's incredible.
It is.
Now, a lot of the research we're looking at focuses on Aedes aegypti mosquitoes.
Right.
Why mosquitoes and why this species in particular?
Well, for one thing, Aedes aegypti is a major vector for some really nasty diseases.
Oh yeah, like dengue and Zika.
And controlling those diseases is a huge global health priority.
Makes sense. And mosquitoes also have a relatively short generation time. So they're ideal for studying gene drive dynamics because you can see the effects pretty quickly.
That's true.
But the communications biology paper points out that there are tons of other insect pests out there that could potentially be targeted with SEMs.
Definitely.
So while these mosquitoes are like a really important model organism, the long-term vision is to develop tools that can be applied to a much wider range of species.
Right. An ecological context.
And that's where mathematical models come into play.
Absolutely.
We need to be able to predict how these gene drives will behave in the real world. We can't just release them and hope for the best.
No.
So the PLOS genetics paper talks about using deterministic models to predict the spread and persistence of SEMs.
Yeah.
They've taken existing gene drive models and modified them to incorporate things like the probability of transgene elimination.
Exactly. They're adding another layer of complexity to these models.
Right, because now you have to account for the self-destruct mechanism.
And it's not just about the probability of elimination. There are all these other factors that can influence how effective the CES is.
Oh, like what?
Well, things like the fitness cost of carrying the gene drive.
Like if the gene drive makes the mosquitoes less likely to survive.
Exactly.
Okay.
Or the frequency of releases, even the specific genomic region targeted by the nucleus can all have an impact.
It's like a giant puzzle with all these interconnected pieces.
Yeah, and that's why these models are so valuable.
Because they help us make sense of all that complexity.
Exactly.
They can help us predict how these different factors will interact in the real world.
Right.
Wow, we've covered a lot of ground here.
Yeah, we have.
We've talked about the basic concept of self-eliminating gene drives, the science behind that self-destruct mechanism, and why researchers are focusing on these particular mosquitoes while also thinking about the broader potential applications.
Right.
But this is really just a tip of the iceberg, isn't it?
Oh, yeah. There's so much more to explore.
Okay, well then let's take a quick break and then we'll come back and dive even deeper into the actual research findings and what they tell us about how effective these self-eliminating gene drives are.
Sounds good. So one of the things that I find really interesting about this research is how they're using different organisms and mathematical models to refine these self-eliminating gene drive systems.
Yeah, it's like they're trying to find the perfect recipe.
Yeah.
Carefully adjusting the ingredients and testing different combinations.
That's a great analogy.
And one of those key ingredients, so to speak, is the choice of organism. We talked about Aedes aegypti mosquitoes being a primary focus because of their role in spreading diseases.
Right.
But the PLOS genetics paper highlights some work with fruit flies as well.
Yeah.
Fruit flies, why the shift?
Well, fruit flies are like the workhorse of genetic research.
Okay.
They reproduce quickly. They have a really well-understood genome. So they're ideal for studying the intricacies of these gene drive mechanisms.
So it's like using a simplified model to test a concept before applying it to a more complex system.
Exactly.
So fruit flies are like the test kitchen for gene drives. Before you move on to the main course of mosquitoes,
Yeah.
what kind of insights are they gaining from these fruit fly experiments?
Well, they're uncovering some really important details about the factors that influence SSA efficiency.
Okay.
For example, the PLOS genetics study found that the length of those direct repeat sequences we talked about plays a big role.
Remind me, why are those direct repeats so important?
They're basically the guide rails for the SSA pathway.
Okay.
The longer the repeats, the more likely the cell is to successfully remove the transgene during DNA repair.
Gotcha.
But what's fascinating is they found there's like a sweet spot.
Oh.
Increasing the length beyond a certain point doesn't actually improve efficiency.
So it's not just about making the repeats longer. It's about finding that optimal length.
Exactly.
It seems like every aspect of these systems requires such precise fine-tuning.
Yeah, it really does.
And the choice of nucleus is another crucial factor. That's the enzyme that cuts the DNA.
Right.
The communications biology research compared two different nucleuses, ICI and Y2INI, and found that they have different efficiencies in triggering SSA.
Yeah, so not all nucleuses are created equal.
It's like choosing the right tool for the job.
Exactly.
And their research suggests that the choice of nucleus might even depend on the specific genomic region you're targeting with the gene drive.
Yeah, it's a complex interplay of factors.
Which makes those mathematical models even more crucial, right?
Absolutely.
So we talked about how the PLOS genetics researchers have modified existing gene drive models to incorporate the probability of transgene elimination and other factors specific to SIMS.
Right.
But the communications biology paper takes it a step further.
Yeah.
They highlight the importance of spatial modeling.
Spatial modeling.
Yeah, what's that?
Well, it's essentially about predicting how a gene drive will spread geographically. Remember, one of the goals of SIMS is to limit the spread to a specific area.
Right, like you don't want it to just run wild.
Exactly.
Okay.
So spatial models help researchers test different release strategies and predict how environmental factors might influence the gene drive's dispersal.
So things like wind or the movement of animals.
Exactly.
So it's like a virtual map.
Yeah.
Allowing them to simulate how the gene drive will move across a landscape.
That's a great way to put it.
And these models are becoming increasingly sophisticated.
Yeah.
The communications biology paper mentions a software suite called MG Drive E. which allows researchers to simulate not only SIMs, but a whole variety of other gene drive systems as well. We briefly touched on MG Drive earlier.
Right.
What makes this software so special?
Well, it allows for incredibly detailed simulations.
Yeah.
You can factor in things like the genetic makeup of the population, the fitness costs associated with the gene drive, the specific environmental conditions, all sorts of things.
So researchers can run countless virtual experiments,
Yeah.
testing different scenarios, tweaking parameters, seeing how it impacts the gene drive spread.
Exactly.
It sounds like an incredibly powerful tool,
It is a powerful tool,
almost like a crystal ball for gene drives.
but it's important to remember that it's still just a model.
Right.
The research notes caution that the findings from these simulations always need to be validated in real-world experiments.
Of course, the real world is always more complex than any computer program can capture.
Yeah, for sure.
But it seems like these models are essential for guiding research and helping scientists make informed decisions about how to use gene drives safely and effectively.
Precisely.
It's about combining this cutting-edge technology with a deep understanding of both genetics and ecology.
Yeah.
And that brings us to another important aspect of this research,
Okay.
the potential applications of SEMs.
Right.
We've talked a lot about the science, but now I'm curious about how these self-eliminating gene drives could be used in the real world. What kind of problems are they aiming to solve?
Well, the Philosophical Transactions of the Royal Society B paper offers a really good overview of the potential applications.
Okay.
They discuss two main strategies, temporal control and spatial control.
Okay, break that down for me.
Sure.
What do they mean by temporal and spatial control?
So temporal control is basically about setting a time limit on the gene drive.
Okay.
It's like programming a self-destruct timer. After a certain number of generations, the gene drive disappears from the population.
So it's like a temporary intervention.
Yeah.
You use the gene drive to address a specific problem,
Right.
and it fades away.
Minimizing the risk of long-term ecological disruption.
Okay, makes sense. What about spatial control?
So spatial control is about limiting the geographical spread of the gene drive.
Okay.
It's like creating an invisible fence. The gene drive might spread within a defined area,
Okay.
but it's designed to be much less effective or even self-eliminate outside that boundary.
So you're essentially containing the gene drive,
Yeah.
preventing it from running wild across a landscape.
Exactly.
That seems crucial for addressing those concerns about unintended consequences.
Absolutely.
And the paper highlights how mathematical modeling can be used to design these spatial control strategies. Researchers can simulate how the gene drive will spread under different conditions.
Right.
And adjust the parameters to optimize its effectiveness while minimizing its impact on surrounding populations.
Exactly.
This all sounds incredibly promising,
It does.
but I imagine there are still challenges to overcome, right?
Oh, yeah, definitely.
Developing these systems is a complex undertaking.
Yeah.
And there's still a lot we don't know about how gene drives will behave in these complex real-world environments.
That's true.
Okay, then let's shift gears a bit and explore those potential challenges in more detail.
Okay.
What are some of the things that could go wrong, even with these carefully engineered, self-eliminating gene drives? So what are some of the things that could potentially go wrong, even with these self-eliminating gene drives?
Well, one of the biggest concerns is the possibility of resistance evolving in the target population.
Resistant. So like the mosquitoes, for example, finding a way to overcome the gene drive.
Exactly. The Philosophical Transactions of the Royal Society B paper talks about this, explaining how even a single mutation at the target site could make the gene drive ineffective.
It's like trying to fit a key into a lock that suddenly changed shape.
Yeah, that's a good way to put it.
And the communications biology paper points out that this has actually been observed with CRISPR-based gene drives in the past.
Right. Mosquitoes can evolve resistance surprisingly quickly.
So it's like a constant arms race.
It is. Researchers are exploring ways to design gene drives that are less likely to trigger resistance.
Okay, how?
Well, one idea is to target multiple genes simultaneously.
So instead of one lock, you have several.
Exactly, making it much harder for the organism to evolve a way to bypass them all.
Makes sense. Are there other potential downsides beyond resistance?
Another worry is unintended spread. Even with these spatial control measures in place, there's always a chance that a gene drive could jump to a non-target population.
Like wind carrying a modified mosquito beyond the intended boundaries, or hitching a ride on a ship or something.
Precisely, and that's why the Philosophical Transactions of the Royal Society B authors really emphasize the need for robust ecological risk assessments before any field trials are conducted.
So, like, really thinking through all the potential consequences.
Yeah, they even suggest that in some cases a reliable self-elimination mechanism might be a prerequisite for even considering a release.
It's like a safety net.
Right, even if the gene drive escapes, it's programmed to disappear after a certain amount of time.
Reducing the risk of long-term harm to those unintended populations.
Exactly, but of course it's not a foolproof solution.
Right, nothing ever is.
And that's why those mathematical models are so important.
Yeah, we keep coming back to those.
It's because they allow us to simulate different scenarios and assess the likelihood of spread under various conditions.
But the communications biology paper also mentioned the limitations of even the most advanced software. They cautioned that real-world validation is always necessary.
Absolutely. Models are incredibly useful tools, but they can only take us so far.
Right.
Real-world ecosystems are messy.
Yeah.
They're complex, and there will always be unforeseen variables.
Like unpredictable weather patterns, the movement of animals, the interactions between different species.
Exactly. All things that are very difficult to capture in a model.
Which is why they're emphasizing a cautious, step-by-step approach.
Definitely.
Lab experiments, carefully controlled, field trials, ongoing monitoring. All of that will be crucial for ensuring that these self-eliminating gene drives are used safely and responsibly.
I think that's a key takeaway from all of these sources.
It is. We're dealing with a really powerful technology here.
Yeah.
And we need to proceed with a lot of humility and a deep respect for the complexity of nature.
I agree. I think the sources you shared all point to a real commitment within the scientific community to transparency.
And to a thorough consideration of both the potential benefits and the risks.
That's reassuring to hear.
It is. It seems like they're approaching this with a healthy dose of caution.
Yeah, it's not just about pushing the boundaries of science.
Right.
It's about using that knowledge responsibly to address some of the world's most pressing challenges.
Finding that balance between innovation and responsibility.
Well, this has been an incredible deep dive
It has.
into the world of self-eliminating gene drives.
Yeah.
We've covered so much ground,
We have.
from the intricate science behind them to the potential applications, the challenges that lie ahead. It's clear that this is a field with enormous implications for the future.
It really is. I'm excited to see what the future holds for this technology.
As are we all. A huge thank you to our expert for guiding us through this complex topic.
My pleasure.
And to you, our listener, we hope this deep dive has given you a better understanding of this fascinating and potentially transformative technology. Until next time, keep exploring.